welcome everybody. First of all, I would like to acknowledge the organizing committee, Mara, Ana Souza, Márcia Neves, Marian Colony, Francisca Silva and Rita Teles for organizing this workshop on a very important subject, of course, cancer. And thank you for showing that materials science is everywhere. It's and in this case also chemistry, and of course cancer needs other weapons to be tackled, but it's all materials is also part of the provides tools also for fighting this problem. Well, having said that, it is my great pleasure to introduce the first speaker, my friend João Conde. João is one of our main scientists in nanotechnologies and microsciences. João is at present president of the Department of Bioengineering of Instituto Superior Técnico. He's also director and group leader at INESC, Microsystems and Nanotechnologies, and where he directs the thin film MEMS and biomems research group. João did his PhD in Princeton University in electrical engineering, and then he did a postdoc in what is a legendary place, IBM at Yorktown, USA. He's one of our main scientists also in the field of microfluids, with very considerable experience in modeling, design, microfabrication, characterization of lab-on-chip systems for integrated biosensing of biomarkers, toxins for cell chips. And this talk will, of course, be on this topic, lab-on-chip platforms for biological analysis. João, it is a great pleasure to have you here. Thanks for participating, and the floor is yours.
Thank you very much. It is very nice to see so many familiar faces. So, can you see my slides in the screen?
No.
Good. So thank you for the invitation, and I'll start right away, and perhaps I'll start with a small provocation. So I would like to claim that the ultimate engineering achievement in the early 20th century has to do with microfabrication. So at this moment, we can do, and we have examples of this around us, for example, in our cell phones, that the microelectronic chips have reached minimum feature sizes below 10 nanometers and more than 1 billion devices on each chip. These are almost quantities that have almost an astronomical dimension, in the sense that they are hard to fathom. 10 nanometers are 2 proteins side by side, and the billion devices, is a comparison with the number, the population, 1,000 million people. And the toolbox to do these chips is the clean room micro and nanofabrication technology. Okay, and in this picture, I show you an example of the interconnect of an integrated circuit. These small lines here, this is an old generation, so these small lines here are about 50 nanometers in the transversal dimension, and then it goes up to a few microns up here, right? So what this technology allows us to do is it's a materials processing technology, and it allows us to sculpt materials. And this is the basis for the lab-on-a-chip devices. Okay. Now this technology is very simple to understand. I only need one slide to explain you. Let's imagine here that I have a substrate, could be, in this case, in this example is silicon, and I want to sculpt the silica that I deposited on the top, for example, by a chemical vapor deposition. So what I do, I deposit this film of silica, then I spin coat a polymer on the top, and this polymer has the characteristic that I can change it chemically by depositing energy. This energy could be a photon, an electron beam, etc. What matters is that I change chemically my polymer, and then I remove the changed part, or depending on the chemical activity of the polymer, the part that was not altered. Okay, so I can pattern the polymer. So this is the second operation. So that we have deposition, patterning, and the final deposition is an etching deposition, which is it's an etching process. It's a mixture of chemical and physical, and allows me to remove the material that is not protected by the photoresist. Okay, so we only need these three operations to pattern materials. The evolution during the last 40 years has been to be able to pattern these materials in smaller and smaller scales. Then what we do is we go around. Okay, we repeat this cycle of deposition, patterning, and etching to achieve our three-dimensional object. The current minimum feature sizes are somewhere between 5 and 10 nanometers. Okay. Now, what we have here at INESC MN, which is quite unique, is the infrastructure to do this process. And some time ago, I'm trying to. Okay. Some of our graduate students made this film. I'm going to see if it works. Yeah, it works. So we have this film. Sorry for the propagandistic aspect, but it gives you an idea of the environment that is required to do this micro sculpturing of the materials. Okay. So it's a clean environment in terms of particles. You need different parts of different cleanliness levels. You can see the substrates, and then this lithography is the operation to expose the photoresist. Okay, and it's possible then to pattern this photoresist and then etch the devices. Okay, so this is quite a specialized environment, like you have also in your labs. It's just different. That is required for electronics, but also for sensors and macro mechanical systems and microfluidics. So you'll see here some examples soon. So you can see a little biochip here being instrumented. Also here you can see the biochip, and you can see here images of the fluids passing in the chip. I'll actually end this conversation with a small discussion about so this is to say that you need this special infrastructure to do these microfabrication systems. Now, if this materials processing technology was developed for microelectronics, but in the last 25 years, spin-offs of microelectronics have been developed. One of them I'm not going to talk about today a lot is MEMS, electromechanical systems. So in this case, it is possible to change slightly the processing techniques to make objects that are three-dimensional. And when they are three-dimensional, they can have movement. Okay? And so this is very interesting for sensors and actuators at the micro and nano scale. Okay. We all have this, for example, in two very important technologies. One of them is in our cell phones. In our cellular phones, we have gyroscopes and accelerometers that are made with MEMS. This is important because of the miniaturization. And essentially, all the motion sensors in our cars, for example, are also made with MEMS. You can have redundancy, you can have higher reliability, etc. Okay. So sometimes when we talk about microsystems, it's the MEMS that we are thinking about. For example, people like to think about a micro doctor some kind of micro robot that we inject in our circulatory system, and it's going to check out if we have some atherosclerotic plate or something like this. Okay. Now, MEMS are important, but for biological applications, the direction has gone in a different way. Okay. And this is what I want to discuss with you, which is this aspect of microfluidic structures. So the microfluidic structures, actually, the concept is relatively simple. If we use this etching process, we can make channels, right? And once you make channels, if you seal your channels with a cover, you can have a fluidic system. Okay? So the idea of microfluidics is to miniaturize, is to use microfabrication to make pipes, pumps, valves, and so that you can miniaturize any fluidic operation that you want. Could be for chemistry, biochemistry, biology, etc. Okay? And so the three main applications that we see for microfluidics, one of them is in chemical analysis and bioanalysis. This is what I'm going to discuss a bit more today. Another application that is being discussed is microreactors. So instead of an analytical aspect, a synthetic aspect. And something that I remember in my classes, I used to have one slide saying, one day we can do cells, tissues, organs, and organisms on a chip. And this has truly exploded in the last 5 years. And there's an enormous activity doing cell cultures and tissue cultures on the chips. Okay? So today I'm going to focus mostly on the biosensing aspect and discuss with you what we need to think about when we think about this. Now, if you do microfluidics, there are a few things that you should know. And they are summarized here in this little, in this little blue panel here. Let me see if I can get the laser. So the first thing that happens is that because you decrease the channel, the Reynolds number in microfluidics is usually very low, so you will always operate in laminar flow. Okay? So this is on one hand good. It's very easy to calculate the velocity fields, the concentration fields in your device. On the other hand, if you want transversal mixture, you have to rely on diffusion. Of course, diffusion tends to be quick because the distances are small, not because it's the same physical phenomena, but the distances are small. But you know, proteins and cells diffuse very slowly. So you need to take into account that there is no turbulence in microfluidics. A second aspect is that you have very high hydraulic resistance. If you remember the Hagen-Poiseuille law, your hydraulic resistance goes with the force power of the hydraulic radius, right? So it's like pulling teeth moving liquids in microfluidics. You have to consider small volumes because the volume scales with the cube of the dimensions, right? So if you decrease the dimension, transversal dimension 100 times, you decrease the volume 1 million times. So it's easy to have nanoliters and picoliter devices. You have large surface to volume ratios, right? So surfaces play a much more important role than at larger scales. So you need to control your surface chemistry and physics, and surface tensions play a very important role. So essentially you use Navier-Stokes like in big objects, but you have to take into account some of these physical aspects, right? So this is an important thing. But when people started discover the possibility of doing microchannels, immediately they started thinking about microanalytical systems for point of use, point of care. Okay, this is a cartoon from Science already in 2002, when they were speculating that you could take a sample from this little kid that is sick with fever. So, at this moment she could not enter she could not enter the hospital. This is a very strange idea. I'm still waiting to have fever, needing to go to the hospital and being told that I cannot enter. And there is a device, miniaturized device that has somehow sequenced the virus that has infected the kid and has sent it to the pharmacy to produce some medicine. Okay? So this is the idea, generically. Okay. And the broad goal, of course, this is from Wikipedia. There is an enormous amount of biomarkers that we will be interested in measuring. Okay, of course, this is very small. We were involved in a project some time ago that was looking at PSA, prostate-specific antigen, which is somewhere here. Okay, this little red thing here is the clinical range of PSA. So there's this enormous aspect. Now, after this enthusiastic approach, I wanted to cool down a bit our expectations. So there is this aspect, again, this is another early speculative drawing on how you can miniaturize central labs into a chip, and for lab on a chip, biodiagnostics, chemical diagnostics, environmental diagnostics, right? And there are many reasons why we would want to do this lab on a chip, okay? And here I have a list. So one aspect, so the point here, I think, is that usually there has to be one good reason. You don't need to have all these reasons, but there has to be one good reason to miniaturize. So in some cases, portability is important. Okay? We need to have it's important to have point of care or point of use or point of impact diagnostics. Okay? For example, to make measurements at home, outside of a clinical setting, or in a remote clinic. Okay? Portability can be important, and it can be the decisive reason. For example, even in the medical consultation office, in many cases it would be nice to have these measurements during the consultation or just before. Like now, so we were just talking before the start that now I'm a regular customer of ophthalmologists, and we all know that when we go to the ophthalmologist before we see the doctor, we run through a series of tests, right? Before we see the doctor. Lab on a chip would allow us to run a series of tests also at the clinical office before we see the doctor and get an immediate expedite diagnostic and etc. Speed can be very important. Okay, the ability to get an analysis very quickly, for example, in cardiovascular diseases and others, is important. The ability to multiplex, to measure many parameters at the same time. Miniaturization is good for this. The capacity, high sensitivity has to be demonstrated, of course. Integration of cellular operations could be important. I'll talk a lot about this in the next few slides, but the idea that the chip can do the sample processing, several aspects of sample cleaning, and then the biodiagnostic is an important advantage. In some cases, just using small volumes of samples or small volumes of reactants can be important if the samples are scarce. And in the end, we need low costs. Okay, so any of these aspects could be a decisive aspect for miniaturization. Now, how do we do this? The simple way is how I describe here. We have to get the fluid in through the channels, right? Then we have to have a biosensing layer with probe molecules that do molecular recognition. Okay. Then we need to have some kind of transduction from the molecular recognition aspect to a physical signal. Then we have to get the information out, and the fluids have to be out. Okay. This is a naive vision, but just this naive vision involves a lot of discussion. Sorry for this complication, but the chemists and physicists, well, we are in an audience of chemists and physicists. You will understand this. What I have here is the microchannel, right? And you have to consider all these things. You need to consider the mass transport. Convection in the axial direction, diffusion in the transversal direction. You need to consider the reaction kinetics, right? The capture of the target by the probe molecule. And this is usually kind of an equilibrium process. Then you need to consider some kind of signal generation for the transduction, electrochemical, optical, mechanical, and you need to consider matrix interference. Okay? So all this has to be considered in this assay. But here I go through some more detail. These are the kind of considerations that we need to discuss when we think about a biodiagnostic. For example, if one of you discusses with us, oh, I want to detect this biomarker for cancer, we need to consider the first thing we think is some kind of molecular recognition, right? I need a probe for the target molecule. So we need to consider if we're going to use antibodies, aptamers, I don't know, some kind of chemical molecule that has a special affinity. But then we need to consider that this molecular recognition object has to be in a certain surface, immobilized, covalently, electrostatically, etc. Then we need to think about the assay architecture, right? Lateral flow assay, microscale flow, blah, 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 nanofluidics, etc. Okay? This is also very important. Then we need to think about the signal transduction, okay? How to get the signal out. And extremely important, how to do the sample preparation. It's rare that we can use a raw sample. Do we need to dilute the sample? Do we need to run through a microcolumn to remove some of the interference, etc.? Okay? So this is perhaps the most important slide of my talk. I wanted to call your attention that all this needs to be considered if we want to develop an assay that is useful. Okay? And this is a necessary condition. It's also not sufficient, but it's a necessary condition. And I think in many cases, many of my colleagues, they are very happy with this very quick, simple picture, but interference, equilibrium aspects, quality of the molecular recognition, etc., are absolutely crucial. Okay? So when we come back to here, we need to have direct use of real samples in integration of sample preparation. We need to get a distant signal out, so sensor arrays, signal enhancement, and what is really difficult is to make something that is easy to use. Okay? In the end, the user, the doctor, the lab technician, they couldn't care less about the technology involved in these devices. They want to put the sample in and get a result that has statistical reliability out. And so there's a lot of engineering, there's a lot of chemistry, there's a lot of physics involved, and all these, any of these points can be an important obstacle for the success of the operation. Okay, so this is a bit I'm going to go through two examples to finish my talk, but I wanted to call your attention that you miniaturize the fluidics. We have absolute control over the velocity fields and concentration fields. We can know the chemical kinetics, but we still need to, what we really want is a from sample to answer system, which in a way to making things look easy is very complicated. So now to conclude, let me give you two examples. Unfortunately, recently we have not done cancer biomarkers, but I think by now you understand that we're talking here about a platform that has to be tuned to a specific application. I'm talking here about the detection of infliximab in blood for antibody therapy management. This is a collaboration with João Conde in the Faculty of Pharmacy here at the University of Lisbon. So in some diseases, people produce a lot of a pro-inflammatory cytokine called TNF-alpha, and one of the therapies is to introduce this antibody, an antibody called anti-TNF-alpha that complexes with the TNF-alpha and decreases the inflammation. And it's very important to monitor the quantity of this anti-TNF-alpha. One of the reasons why it's important to monitor is because some people produce anti-drug, anti-TNF-alpha antibodies. Okay, which should also be monitored. So the question here is, can we make a microfluidic biosensor to detect and quantify infliximab and, if possible, the anti-drug antibodies? And the idea is we should be able to monitor this directly from blood at the doctor's practice. Okay. And so the first thing is to establish a molecular recognition idea, and this is what we did on this side. So what we do is we have a channel and we immobilize on the surface TNF-alpha. Okay. Then we run the sample, and if there is infliximab in the patient's blood, it gets trapped by the TNF-alpha. And then we run an anti-IgNG, because this is an IgNG, an anti-IgNG antibody labeled with HRP, which complexes to the infliximab. And then we use chemiluminescence to see the signal. Okay. So if there is no infliximab, this antibody will not connect, and we have no signal. To detect the anti-drug antibody, we will immobilize the infliximab on the surface and capture. Okay. So you can see here the chemiluminescent signal of the infliximab, and just to show you here, we use a simple channel. This is very simple. So we have this is the calibration curve for the infliximab, including in we dilute plasma by 100 times, and here we can see in white the standard ELISA tests, and in black the conventional. This is the conventional ELISA, the microfluidic ELISA, and we can see that in about 10 minutes, well, we can do these measurements in diluted plasma. Okay, so we can detect the infliximab at clinical levels. And we are trying now to measure directly from blood sample. But you see that we established the reaction, the sample preparation technique, and in this case we use optical transduction. The other example that I wanted to show you is a microfluidic system for bacterial detection that we are developing, and we are not concluded. But the idea here is that we would collect. With a nasal or rectal swab, the sample, we should do the cell lysis, do the DNA purification, the DNA amplification, the specific detection, and the integration of the whole thing. So the idea is to have a chip that will start with a swab, and come up with the detection of, in this case, bacteria resistant to antibiotics. Again, you see, this is a very generic platform. I'm only going to show you here what we do, just give you a flavor of the bacterial lysis. So here you can see the design of our chip. The lysis solution and the bacteria suspension enter here. They are mixed. They go around lysing. Then we put a buffer solution to stop the lysis, and we capture the DNA here in chromatography beads. So we have here a mixer to help the mixing in this region here, macro-patterned. We capture the DNA in Q-saffrose beads, which have a quaternary amine. Okay. And at this moment, we then using evergreen, which is a fluorescent intercalator, to detect the DNA. Okay, so you can see. When we don't have bacteria, if we have PBS or lysis solution, we don't get anything. If we have DNA, we get signal, and then if we lyse the bacteria, we can also get the signal. So here, this is the first point. This is the bacterial concentration, and this is the detection level. You can see we can detect Staphylococcus aureus at these concentrations and E. coli at this concentration. Now, this is the direct detection of the DNA after lysis. Of course, what we are doing now is we have also developed that is already an amplification process, isothermal amplification process on the chip, and the detection process optical. What we are working on is in this case, on the low temperature denaturation of the captured DNA, so that we can amplify. But hopefully in the next few months, we'll have a chip that does all these steps. Okay? And the point here that I wanted to make is that you can have something that incorporates multiple steps. In this case, lysis, DNA capture, denaturation, amplification, and detection. To be able to do a high sensitivity analysis in a short time in a portable platform. So to come to the conclusions, I hope not so much convince you, but I suggested that the individual operations of standard chemical, biological, and biomedical analytical labs can be miniaturized using microfluidics. There has to be a reason, right? Economical or technical reason. That microfluidic microsystems can be used for low-cost, fast, sensitive, multiplex point of impact biodiagnostics. But they need to be easy to use. So the ease of use, clinical range sensitivity, and limited or no sample preparation are essential for applications. Okay. And before I really finish, I just wanted to show you here some of our work with cell chips, and what you see here. These are 6 chips where we're growing colon cancer stem cells, exactly for the development of therapies against these cells. This is a collaboration with people also in pharmacy in Lisbon. This can be done, but it's a different story, right? It's a different story from biodiagnostics. So just to finish, so our group works on MEMS, lab-on-a-chip biosensing systems, and cell chips. So I told you a little bit about the lab-on-a-chip biosensing systems. And of course, I wanted to thank you for your attention, and again mention that we are always open for discussion and collaboration with our colleagues all over the country. So thank you very much.
Thank you, João, for your enlightening talk. Very simple. I think everybody could understand you, and thank you for keeping the time. Very well in time. Are there any questions? People should not be shy. They are hiding behind technology.
João, there is two people that raise their hands. Okay, three people, in fact.
Okay. Yes, there is. I see Anna.
Yeah.
Anna, please.
Hi. Thank you so much for your talk. It was very interesting. I'm totally out of this area. I'm a neuroscientist at the University of Benin-Interior, and I have a question for you, João. Can your technology be used as a drug administration device that we could actually implant under the skin for the controlled diffusion of drugs?
Yeah, there's a lot of work on the aspects. So the main direction of work has been on devices that include microneedles. Okay, yeah. For example, so this is quite. There's a lot of work on this. So that is not so much implanted as like a surface type of delivery. There is early work by Whitesides from Harvard on a self-contained device that had several cavities, and then using the dissolution of membranes did the drug delivery at specific times. The problem I see there is how to give the instructions to the machine, right? Because you are implanting, maybe you need some kind of antenna, an energy source. It becomes challenging. So in principle, it's possible, but miniaturizing to that level and having an active device, right, that you can say, now deliver this, then deliver that, is not so easy, right?
Yeah, I understand, because I think your technology have two very important points. I mean, several, but in my area. We can actually, if I understood right, you can detect, and then it is possible to release, right? Yeah. I was wondering, for instance, for the Parkinson disease patients that need to have a very tuned delivery of dopamine. If your device can detect dopamine, and once the levels are in critical points, give some kind of instruction to the device to deliver more dopamine to the system. Yeah, this was some kind of idea. As I told you, I'm totally out of the area. I don't know if it's possible. Obviously.
I would say that the detection tends to be easier than the delivery. Remember that, for example, for the diabetics, I remember a couple of years ago when we still had conferences, there was an invited speaker that more than 30 years ago was trying to do the device, an implanted device for the delivery of insulin, right? And there are all this. The main issue is actually a materials issue, an issue of biocompatibility, right? That to avoid some kind of aspects of infection. But in principle, these devices, if you could have the energy source, some kind of MEMS device that could allow the delivery. But these are not simple devices to make. No.
Yeah, I understand that.
These are not simple devices, and particularly, if it's if it's if it's not, if it's in deep tissues in our, particularly in the brain, right?
Yeah, that's true. It's even worse. Anyway, thank you so much. I really enjoyed it.
Thank you, João. There is also, I think, Ana Souza would like to put a question.
Yes, if I have some opportunity. Thank you so much. It was brilliant presentation, and for me that it's not from the area, I could understand a lot better about this. I have one question, maybe about, you told about the cell that you are able to do on lab-on-chips. What about organs? How far are we? I know that there are already some lab-on-chip organs, but how far are you from this? And my question is because we have very strict directives nowadays to reduce animal testing, and we do need a lot of lab-on-chip organs. So is something that you are working on right now? Can we expect a collaboration in the future for artificial lung, for example?
Yeah, well, there, it's a very good question. There, I would say that no, maybe somebody else could not share my opinion. I think there the problem is not so much the technical problem, because we have models, of device structures that can mimic most of the tissue-like interfaces, like a lung, like make a porous membrane. You put some kind of fibroblast, and then at the other cells you can make movement. I think the main difficulty is a difficulty of communication between, let's say, the technological people and the biomedical people. Because, for example, myself, but I think, I'm in the Department of Bioengineering. I've been working with biologists and bioengineers for decades, but I don't know enough about the the whole architecture of the organs to do a device that makes sense, right? On the other hand, the people on the medical side and biology, biomedical side, have to be able to somehow abstract from their organ the essential features that we need to have in a device, right? And so I think this communication is very difficult, right? Mm-hmm. And this is, I would say, then later on so on the technical part, I would say that the more challenging thing will be the vascularization, right? So the vascularization we'll need to talk a lot about. But generically, I think it's more to know exactly what you have to do to mimic what you want to see, right? What is the simplest thing? Rather, like, I think biologists and biomedical people like to complicate, we need to kind of depurate, depurar in Portuguese, right? Mm-hmm. The essence, so that we can have something that is workable, right? We cannot imitate the complexity of a lung, but we can find the moduli, that can kind of reproduce the features, and then you can do it. Lung, liver, all these things.
Yeah. Maybe brain with Ana Clara in the future. But
these are things that require time and money, right? And investment, a lot of effort. It's not some experiment. Like I would say, it's much easier to do a biosensing device where we know, I want to detect this molecule in this environment. We discuss a little bit, we see what is the standard, and we try to imitate. Now, making a cell chip that has a function of an organ, I think it's a kind of a 5-year program.
Yeah, kind of a marathon. Yeah. Thank you so much.
Okay, João, we still have a couple of minutes. What do you think is the main limitation of the lab-on-chip technology, let's say? Microchip. Yeah.
I think at this moment, so what you have at this moment in the market is, I think it's the easy to use. Okay, the ability to use it inexpensive, make it easy to use, because the chips that are easy to use are all, are most of them the lateral flow assays with membranes, right? With capillary membranes. But these devices are one-step assay, So they don't allow all this amplification and purification processes that we have in a standard laboratory. Being able to have the easy to use, reasonably priced device has been a very big challenge. Okay, there are half a dozen companies in the world. One of them is Portuguese. Actually, the BioSurfit has been doing a lot of effort. They use a lab on a CD. Okay? These are not the cheapest assays, but they have a machine. You use a lancetta, you get a drop of blood, you put it there, and after 10 minutes you have your solution, right? So I think ease of use, and of course a system that has some money for diagnostics.
Of course.
I think this is the main issue.
Thank you, João. I clap my hands.
Thank you all.
So we move on, and now we have Joana Rodrigues.
Hello, good morning.
Olá, Joana. Joana will give a talk on ZnO-based transducer platform for biosensing applications. Let me introduce Joana quickly. Joana is a junior researcher at INESC in the physics department of University of Aveiro, the second associated laboratory in Aveiro dealing with materials. Her work focuses on the characterization of wide band gap semiconductors, namely nanostructure semiconductor oxides to be applied in optoelectronics and sensing devices for detection of biological analytes relevant for health and environment. Joana, welcome. And
hello, thank you.
Yeah, and the floor is yours. You can start your talk.
Thank you.
Okay.
Can you see my slides? Okay, good morning. Thank you for the introduction. My name is Joana Rodrigues, and I am a junior researcher at the physics department in the University of Aveiro. And first of all, I would like to thank the workshop organizers for inviting me and giving me the opportunity to be here today, presenting the work that we have been conducting in the biosensing field. So in this presentation, I will talk a little bit about our research project that consists on the development of zinc oxide-based transducer platforms for biosensing applications. Here is the outline of my presentation. I will briefly explain the motivation for proposing this project, present the NanoBiosense project itself and its main objectives. Then I will show you the main materials that we have been applying as transducers in our biosensors and some of the results that we already attained, focusing on the detection via the luminescence signal of zinc oxide nanostructures. Specifically, I will present two case studies, one devoted to the sensing of the HGC hormone and another concerning the detection and quantification of hydrogen peroxide. Finally, I will finish with some remarks. So, the continuous improvement of healthcare methodologies are one of the most admirable objectives challenging the scientific community nowadays. Therefore, the development of biosensors capable of monitoring disease biomarkers is of paramount importance, since there is a urgent need for tools that allow the early detection and monitoring of health conditions, providing prompt results with high sensitivity and accuracy and at a low production cost. Moreover, such systems should be easily handled and not require skilled personnel to operate them in order to allow their spread worldwide. Indeed, the progresses in nanotechnology in the last years have conducted to remarkable achievements in the biosensing devices, namely by employing nanomaterials in their fabrication. Compared to their bulk counterparts, nanomaterials evidence unique properties, promoting a higher sensitivity to biological targets and enabling a fast detection of multiple analytes, which is desirable in developing biosensors. Indeed, the ability to tune the material properties at the nanoscale, particularly its morphology and surface area, is crucial for the fabrication of transducer platforms, since these properties have a high influence on the detection signal outcome, as well as on the amount of the bioreceptor that can be immobilized on one device. And since this bioreceptor is the element that confers specificity to the sensor and that will determine the number of availing binding sites for the analyte, the selection of a suitable transducer that maximizes this immobilization is critical to obtain an adequate biosensor. Therefore, with the NanoBiosense project, we aim to develop zinc oxide-based nanostructures to be used in the fabrication of optical and electrochemical devices for the detection of analytes with clinical and environmental interest. To do so, we are producing and characterizing zinc oxide nanostructures and zinc oxide nanocarbon composites to select the ones that evidence the desirable properties as transducer elements for our biosensors. And these materials are being produced by innovative and sustainable laser processing techniques that we have available at our lab. With such approach, we expect to contribute with high-quality materials that can be incorporated into easily fabricated and industrial feasible sensors. These transducer platforms will be then modified to attach the proper bioreceptor for the analytes that we aim to probe. And for this biorecognition part, we are mainly using enzymes or antibodies to recognize and interact with the target species, taking advantage of the high specificity provided by them. In fact, to be able to change the target species only by varying the biorecognition, in fact, we expect to be able to change the target species only by varying the biorecognition layer that we place on our transducers. Regarding the signal used for the detection, the unique photoluminescence properties of zinc oxide nanostructures offer a potential transduction platform for sensing, which can be coupled to the standard electrochemical response, especially when zinc oxide carbon composites are used. Thus, we want to produce biosensors capable of combining both optical and electrochemical transduction in the same device. The combination and cross-section of these two sensing mechanisms is expected to lead to a wider range of detection, higher specificity, and even provide a counterproof analysis using the same device and sample by performing distinct measurements in the same sensor. So we believe that achieving this purpose will represent an important contribution to the biosensing field. We choose to use zinc oxide and carbon-based materials in our sensors, since both presents interesting properties for such application. In the case of zinc oxide, we highlight the intense luminescence at room temperature, the biocompatibility, and the high isoelectric point, which is suitable for the immobilization of some proteins which have a low isoelectric point, as is the case of the enzymes or some of enzymes or antibodies. Additionally, the ability of synthesizing this material in the wide range of morphologies with a high and tunable surface area by low-cost techniques is also an important advantage. In our case, we are using a technique called laser-assisted flow deposition to produce our zinc oxide nanostructures. This is a method that has been developed by our research group in the last few years and that exhibits a high yield for the production of micro and nanostructures. Well, this technique enables us to produce zinc oxide structures with different morphologies and sizes, having high crystalline quality and excellent optical properties, which is vital if we want to use their optical signal for transduction. These CEM images show the zinc oxide tetrapodal structures that we are using in our sensors. These tetrapods present branches with a high aspect ratio, providing a high surface area, which is desirable for sensing applications. For the carbon material, we are using laser-induced graphene. This can be seen as a three-dimensionally highly porous graphene foam that is produced by radiating a polyamide layer with a laser beam, resulting a high local temperature that induces a photothermal process. And the rearrangement of the carbon bonds will lead to the production of few-layer graphene with a high porosity due to the gases releases during the process, as you can see in these CEM images. The interest in using laser-induced graphene composites arises from the fact that combining these two materials can lead to more advantaged properties and enhanced sensing performance, and we want to explore this possibility. Hence, several approaches were employed in the production of the composites. Among those, two were already selected based on our preliminary results. The first corresponds to the mixture of the zinc oxide tetrapods that I showed you before, with the laser-induced graphene scrapped from the polyamide substrate. This approach has the advantage to allow a more accurate control of the rate ratio of both components, enabled to somehow adjust the final properties of the composites in line with the type of signal and analyte that we are targeting. The other method is the direct growth or deposition of zinc oxide into the laser-induced graphene that remains on the original polymer. In this case, we choose the synthesis parameters in a way that prevents the full polymer degradation to keep a thin and effective polyamide layer that provides flexible mechanical support to the device prepared in such way. And to complete our sensors, the biorecognition layer is immobilized onto the surface of the transducer material to confer the selectivity and specificity for the target species. And of course, an additional passivation step is employed after the immobilization of the biorecognition layer, which is required to block non-specific binding sites and the direct interaction between the transducer surface and the analytes. So this is the basic architecture of our prototype sensors. Now, moving on to the detection part. Despite the enormous progresses in the field during the last years, the majority of biosensors are still based on electrochemical mechanisms. However, alternative approaches have been raising attention, and one of them is using the optical signals. And besides their high selectivity and specificity, optical sensors allow to measure the properties of the materials without the need of electrical contacts, providing remote interrogation and detection. So in this presentation, I will focus on one of these optical methods. And even though the direct photoluminescence from the transducer is not widely explored for detection, this is an excellent way to assess the properties of the materials, in particular their surface, which is where the sensing reactions occur. And so it is very useful to analyze, this technique is very useful to analyze the bio-nano interface between the transducer, the bioreceptor, and the analytes. Furthermore, since the luminescence signal is very sensitive to small modifications in the semiconductor surface, it allows to discriminate small target concentration changes, providing high sensitivity and selectivity to the sensors using such signal. Therefore, the change in the luminescence response can be used as indicator of the presence of the target substances. Ideally, a signal change, as for instance, the intensity or the spectral shape variation after its interaction with the analyte should correlate with the concentration of that analyte in a way that a calibration curve can be defined. And this is indeed our approach to achieve optical sensing in our samples. So now I would like to focus on two of the works dedicated to the use of the luminescence signal of zinc oxide to assess the presence and concentration of two different analytes, the HGC hormone and hydrogen peroxide. Starting with the detection of the HGC hormone, this analyte was chosen since it is a very well-known protein with a wide variety of functions in the human body, thus constituting an ideal platform for a proof of concept platform for the evaluation of the feasibility of a biosensor based on the luminescence of zinc oxide tetrapods. Hence, to produce our biosensors, we deposited the zinc oxide nanostructures onto adequate substrates and functionalized them with anti-HGC antibodies that are specific for the type of HGC that we aim to probe. In this case, the antibody was immobilized via simple physical absorption procedure, which is favored by the difference in the isoelectric point of zinc oxide and the antibodies. Then the samples were passivated with bovine seroalbumin to avoid non-specific interactions, as I mentioned previously in the first slides. Moreover, samples prepared only with BSA and without any antibody or with one non-specific for HGC were also prepared to act as control samples for the detection measurements. And the sensing evolution tests were conducted by placing a drop of each concentration of the analyte solutions onto the active area of the transducer and let to incubate during 20 minutes under ambient conditions. After this time, the samples were watched to remove the HGC that was not linked with the antibodies and left to dry in air before recording the PL spectrum. This procedure was repeated for hormone concentrations up to 100 nanogram per milliliter and for both sensors and control samples, keeping exactly the same analysis procedure in order to be able to compare the results. To assess the sensing ability of our sensor, we monitored the photoluminescence signal for each concentration and then evaluated the change triggered by the reaction between the bioreceptor and the analyte. The analysis of the resultant spectra revealed that in the case of the samples functionalized with anti-HGC, the overall intensity decreased with decreasing the HGC concentration, particularly in the case of the UV region. This decrease in the PL output was attributed to charge rearrangement at the surface of the semiconductor upon the formation of antibody-antigen link, leading to a decrease in the surface recombination path of the semiconductor and consequently to a reduction in the overall intensity of the signal with increasing HGC concentrations. On the other hand, the control samples, which did not have the specific bioreceptor for AGC, did not show any significant variation in the signal intensity or spectral shape. And these minor changes in the PL signal for the control samples indicate the absence or the negligible non-specific interactions between the zinc oxide surface and the target analyte. And this is particularly interesting in this case, where I show you a sample functionalized with anti-E. coli antibodies, which were not specific, of course, for AGC. And we see that our sensors are indeed specific for our analyte. Here are the response curves as a function of the AGC concentration for 2 sensors and 2 control samples. And in this case, the response was obtaining considering the ratio between the intensity of the UV emission for the different AGC concentration tested and the initial intensity prior incubation. This signal was chosen since it was the dominating emission and one that evidenced more pronounced changes, as you can see here. These results illustrate the reproducible behavior of the tested sensors and the distinct behavior observed for the control samples. The second figure shows the average sensor response for sensors tested, plotting the AGC concentration in a semi-log scale in order to put in evidence the behavior for lower concentrations. As you can see, AGC quantification is fairly possible in the range between 5 and 50 nanograms per milliliter using these sensors, and a saturation of the signal occurred for higher concentration values, which can be explained by considering that the great majority of the antibodies are already connected to the antigens and no more free binding sites are present to interact with further increasing the AGC concentration, thus imposing an upper limit for the detection. Thus, we believe that by adjusting the antibody concentration, we can change somehow the detection range, and therefore further studies will be conducted to clarify this hypothesis, changing the antibody concentration as well as increasing the range of the AGC concentration tested. Even though further optimization is needed in this type of sensors, we believe that these results indicate the adequacy of this material to be employed in the fabrication of sensing devices, and that the same principle observed here can be applied to other immunosensors to prove other relevant analytes for which there is a lack of adequate solutions in the market. In fact, at this moment, we are exploring the same approach for other analytes, namely the ones showed here, and we want to verify if similar changes in the PL signal occur in order to validate this sensing mechanism. The next case study that I would like to show you is the detection and quantification of hydrogen peroxide. As you know, this compound is a common subproduct of many reactions, namely the oxidation reactions involving enzymes, which are in the basis of most of the enzymatic sensors. Therefore, it is also of great interest for us to be able to detect and quantify it using the luminescence signal. To evaluate this possibility, we conducted a series of measurements where we immersed zinc oxide samples in quartz cuvette containing hydrogen peroxide with concentrations from 0 to 100 millimolar. And we saw that pronounced changes in the spectral shape of the PL emission with increasing the hydrogen peroxide concentrations could be observed, as you can see here. Namely, the intensity of the blue band starts to increase with increasing hydrogen peroxide concentrations, dominating over the green one for concentrations above 10 millimolar. This picture here shows the emission perceived by the naked eye for the sample immersed only in ionized water and after being placed in the solution containing 100 millimole of hydrogen peroxide. This blue emission was proved to be originated from the presence of the hydrogen peroxide, whose presence also affected the intensity of the UV and green emission of zinc oxide. Plotting the ratio between the peak intensity of the blue and UV emissions, we can see that between concentrations of 5 and 100 millimole, the trend is fairly linear. Therefore, the changes observed in this signal can be exploited as a possible transduction mechanism for assessing the concentration of this chemical compound. Our next step is to take advantage of this behavior for the indirect detection of analytes as glucose or cholesterol, which rely on catalytic reactions in the presence of the respective enzymes, leading to the formation of hydrogen peroxide. Since we are probing a subproduct common to different types of analytes, and the enzyme immobilized on the transducer surface will be capable of recognizing only the specific target substrate, we expect to be able to tune the specificity of our enzymatic biosensors by only changing the bioreceptor using the same type of sensing platform and signal processing, in line with what was mentioned for the case of the immunosensors. So, to conclude, I hope that I gave you a broad overview of our project. Our main goal is to develop sustainable materials to be used as transducers in biosensing devices. These materials need to have suitable properties to allow us to combine both optical and electrochemical transduction in a single device. And with the selected transducers, we intend to have a sensing platform that enables the tuning of the target analyte by choosing the appropriate biorecognition element. Moreover, as different approaches are being applied to the transducer preparation, we can somehow select the most adequate ones for the type of transduction signal and or analyte that we are targeting. Regarding the detection using the luminescence signal of the zinc oxide nanostructures, we proved that it's possible to correlate the changes in the luminescence spectra with the presence and concentration of the analytes in contact with the sensor, thus demonstrating the potential of the prepared structures to be used in optical-based biosensors. In parallel with the work that I just show you here, also electrochemical detection tests are being conducted, showing also promising results. So finally, I would like to thank to my research group for all the help and support in all the work that I presented here, and to the Portuguese Science and Technological Foundation for the project funding, and of course to you for your attention. Thank you.
Professor, está sem som.
Oh, sorry. Thank you, Joana, for keeping in time. We are not having questions at this point, if I understand well, the organization. So we will have, at the end of the second talk, some questions. And now Matheus Pereira will give us a talk on improvement of cancer biomarkers detection using ionic-liquid-based aqueous biphasic systems for human fluids pretreatment. Matheus is a postdoctoral research associate at CICECO, Department of Chemistry. His research includes, or he has interests in the use of ionic-liquid-based aqueous biphasic systems as pretreatment strategies of human fluids, aiming the concentration purification of cancer biomarkers towards improved cancer diagnosis. Matheus, the time is yours. Could you please go ahead?
Good morning, everyone. Thank you, Professor João Rocha, for the presentation. And also, I would like to thank the organization committee for the invitation for the presentation. And as you can see, the title of this work is the improvement of cancer biomarker detection using ionic liquids-based aqueous biphasic systems for human fluids pretreatment. This presentation will be divided in 3 parts: a brief concept about the problem that we want to overcome, and some details of achieved results about the application of ionic liquids-based aqueous biphasic systems for human samples pretreatment, and finally some conclusions and some trends in this research field. As Ana Souza said in the beginning of this workshop, cancer is the second major cause of death worldwide, with around 9.8 million deaths recorded in 2019. According to the World Health Organization, cancer was the cause of 1 in 6 deaths. The future trends disclose an even worse scenario, with over 20 million new cases expected in 2005. Prostate cancer is the second most common type of cancer in men, representing 17% of the cancer-related disease in males. It is the 5th leading cause of cancer death worldwide. And most of the cancers, including prostate cancer, are the result of a combination of genetic predisposition, lifestyle, and exposure to environmental agents. The change of a normal cell into a neoplastic states involve the acquisition of several capabilities that enables them to become tumorigenic and then malignant. The hallmarks of cancer are constituted by 6 biological capabilities that emerge during the complex development of human tumors. Among them, the induction of angiogenesis constitutes an important cancer hallmark to highlight. Since cancer tumors demand nutrients and oxygen to grow, they require blood vessels. The tumor angiogenesis process occurs by the development of vasculature through creation of a new and spread of vessels. The connections between the cancer tumors and blood vessels is responsible for the exchange of biomolecules between the tumors and the bloodstream. Thus, the metabolic products associated with malignant cells can be found in the human fluids. These biomolecules, which are usually expressed in the presence of cancer, may then be used as a cancer biomarkers. According to the National Cancer Institute, cancer biomarkers are molecules with biological activity or expression that are present in human fluids such as saliva, urine, tears, and serum, being their levels indicative of pathologic conditions. Advances in genomics, proteomics, and molecular biology allow it to identify several biomarkers that are now used as a tool for early cancer detection. Cancer biomarkers include DNA, microRNA, enzymes, proteins, and other metabolites. The prostate-specific antigen PSA blood test is still the most used method due to a strong correlation with the prostate cancer. In the recent years, new biomarkers have been studied and implemented in the clinical trials. Several studies suggest that the serum lactate dehydrogenase LDH levels in patients with solid tumors, including prostate cancer, are correlated with adverse prognosis. Thus, the concomitant evaluation of PSA and LDH in the same patient will allow to achieve a better prostate cancer diagnosis and prognosis. So, several techniques have been applied for a cancer biomarkers detections and quantification. The choice of these methods is obviously dependent on the type of biomarkers to be quantified. For proteins biomarkers, for example, the most commonly used methods are ELISA, high-performance liquid chromatography, electrochemical and optical detection methods. In general, all cancer biomarkers quantification methods in human fluids require samples pre-treatment in order to eliminate the high amount of other non-target proteins and metabolites, allowing the accurate diagnosis. So, aqueous biphasic systems can be seen as a viable method for the extraction, concentration, and purification of selected cancer biomarkers from human fluids with a significant advantage over conventional methods. In the middle of the last century, aqueous biphasic systems, or ABS, was proposed by Albertson as an effective separation and purification platform for the add-value compounds. Aqueous biphasic systems fit within the liquid-liquid extraction technique and are typically composed of non-volatile pairs dissolved in aqueous media, such as two polymers, a polymer and salt, or two salts, which above given concentrations undergo in a liquid-liquid mixing. However, the wild performance of these systems have been blocked by the limited relative difference in polarities between the two phases. More recently, ABS composed of ionic liquids were proposed by Rogers and co-workers, representing an outstanding alternative to the traditional ABS. Nevertheless, one of the most important features of ionic liquids arise from the tailoring ability resulting from a cation and anion combination, and this properties is further extended to the ionic liquid-based aqueous biphasic systems, promoting remarkable advantages compared to the traditional polymer-based systems. So, based on this, our object was in the development of novel strategies for the pre-treatment of human samples for early-stage cancer detection. Firstly, herein we show the applicability of ionic liquid-based aqueous biphasic systems to simultaneously extract and concentrate from concentrate PSA from human urine samples. If the ionic liquid is properly designed, no losses of proteins will occur, not leading to a false positive results and compromising the diagnostic. So ionic liquids formed by a tetrabutylphosphonium cation and good buffer derived anion were synthesized. In order to identify the mixture composition required to perform a liquid-liquid extraction by means of ABS, the respective ternary phase diagram were initially determined, and then the ABS was composed, these synthesized ionic liquids combined with potassium citrate as a phase former. Since the salt temperature, pressure, and pH of these systems are constant, ABS formation ability is a result of ionic liquid nature. As we can see, at 2% of salt, the ability of these ionic liquids to create an ABS following the order. First, we have the anion chess, then mesh, then abject that is very similar to test, and finally tricene with the lowest biphasic area. Basically, ionic liquids with a lower ability to establish hydrogen bond and create hydration complex are more easily salty out. In other words, anions with a lower ability to perform hydrogen bond with water display high capacity for liquid-liquid mixing in ABS. After addressing the mixture compositions required to create an ABS, these systems were evaluated in what concerned the ability to extract PSA. This initial screening on ABS extraction and recovery performance was carried out with commercial PSA in aqueous solution at a concentration around 50 nanograms per milliliter. Two mixture compositions were studied. The first one with 30% of ionic liquids and 30% of salt, and the second one with 30% of ionic liquids and 40% of salt. The extraction efficiencies of PSA in all ABS and mixtures compositions evaluated corresponded to 100%, meaning that PSA is only present in ionic liquid rich phase and no PSA was detected in salt phase. By the characterization of ABS phase diagrams, including a series of timelines that give the composition of each phase for a given mixture composition, it's possible to prepare mixtures with no compositions in defining phases, weights, or volumes. Along the same timeline, by decreasing the amount of ionic liquid added and increasing the amount of salt, the ionic liquid rich phase has a continuously reduced volume with a concomitant increase of concentration factor. By using human urine spike with commercial PSA at a threshold value proposed for a PSA in urine around 150 nanograms per milliliter, in the composition of this ABS, and with a concentration factor of 250, there is the decrease of the volume of ionic liquid rich phase that allows PSA detection and quantification by an equipment with a higher detection limit, such as size exclusion HPLC. The HPLC peak corresponding to the PSA, as we can see in the figure, is not seen in the non-treated urine samples, neither in the salt-rich phase of the ABS. However, a clear peak in the ionic liquid rich phase corresponding to the PSA in the pre-treated urine sample is clearly seen and can be accurately quantified. Remarkably, the size exclusion HPLC proteins profile also reveals the presence of additional protein only in the ionic liquid rich phase of pre-treated human urine samples. The identified peak is BPSA, another form of PSA, and the levels of these different forms of this protein in their respective ratio can be used in a prostate cancer diagnosis. Therefore, the ABS here reported is not only allowed to concentrate PSA, but also BPSA, which can be additionally quantified. Previously, we have shown that ionic liquid ABS can pre-treat human urine, allowing to detect cancer biomarkers. These results lead us to hypothesize if ionic liquid-based ABS can simultaneously deplete highly abundant protein and purify biomarkers from another human sample's serum. To obtain an effective method to purify LDH from human serum while removing the high abundant serum proteins, namely HSA and IgG, we started to investigate two tetrabutylphosphonium-based ABS. A common biphasic mixture composition by 30% of salt and 30% of tetrabutylphosphonium bromide and tetrabutylphosphonium chloride, and 40% of aqueous solution containing LDH or a real serum sample was selected within the biphasic area according to the phase diagram previously reported. In these two systems, LDH is completely purified in the top phase, namely the ionic liquid rich phase, with the purification efficiencies of 100%. However, only in the ABS constituted by tetrabutylphosphonium bromide was able to precipitate the contaminant proteins at interface, with the depletion efficiencies of 100% for HSA and IgG, also in one single step. To optimize the system's efficiency for serum sample treatment, several concentrations of ionic liquids tetrabutylphosphonium bromide from 15 to 45% and salt from 10 to 30% were investigated, and their impact on the LDH partition and HSA and IgG depletion was evaluated. Independently of the salt or ionic liquid concentration employed, the same trends are observed. All composition studies allow the complete purification of LDH to the ionic liquid rich phase without observing the precipitation or denaturation of the targeted biomarker. On the other hand, the complete depletion of HSA and IgG was observed. Aiming to validate the method, an ABS composite of 45% of tetrabutylphosphonium bromide and 20% of salt was chosen to purify LDH directly from serum samples with the simultaneous depletion of HSA and IgG. To this end, several samples were spiked with LDH, and as we can see displayed in the HPLC chromatograms at the ionic liquid rich phase, only LDH is identified, with the absence of LDH aggregates or protein fragmentation. Given that LDH has an intermediate molecular weight between IgG and HSA, remarkably, HSA and IgG were not detected in HPLC chromatograms, thereby not acting as an interferent in this quantification method. As my conclusion, we present that ionic liquid-based aqueous biphasic system can be used in a simultaneous extraction and a concentration and purification process for several cancer biomarkers from human samples. In this work, namely urine and serum, their high performance rests to the possibility of tailoring the polarities and affinities by the proper rearrangement of the ionic liquid ions, and further investigation on the development of effective separation process for cancer biomarkers using ionic liquid-based aqueous biphasic systems towards improved cancer diagnosis and prognosis could be developing using other human samples like saliva, tears, or sweat. I would like to say also thank you for this financial support and for everybody in this workshop. Thank you so much.
Thank you, Tiago, Matheus. We now open both talks for discussion. We have a few minutes for discussion. So who wants to put some questions to Joana or Matheus? Okay, we have Mara, we have Francisca. So Mara, shoot.
Hello, thank you, Joana, and thank you, Matheus, for your talks. They are quite interesting. My first talk goes to Joana, and regarding the work on biorecognition using antibodies, I would like you to explain us better what are the advantages of using your materials and azotical and electrochemical devices when compared to ELISA methods. I mean, in terms of sustainability, time, cost, solvents used, how are they better? Because most proteins and cancer biomarkers are quantified by ELISA in cell plates, so they use low volumes of sample.
Yeah, thank you for the question. Well, we also use the antibodies as the biorecognition element to detect the the analytes, but we are not using the secondary antibody with the fluorophore that leads to the signal that is detected on the ELISA method. In our case, we have our materials that are sustainable materials. We attach the antibody to be the bioreceptor on our materials, and then we incubate also a small amount of sample on that surface. And we use the signal that it's provided by the transducer that is due to the interaction between the bioreceptor and the analyte. And that's it. We don't need any more reagents or secondary antibodies. And also, if we can achieve the optical and the electrochemical measurement in the same device, we will have two different signals that can provide like a counterproof measurement of what we are seeing, avoiding some false positives that are very common in this type of exams.
You improve sensitivity, right? Also, yes, of course, it's more sustainable. But you also, for instance, in the case of the HCG hormone, you do the adsorption, you immobilize the antibody by physical adsorption. How do you control deletion? I mean, then you are adding a drop or a liquid sample, that is your biological sample. Don't you have leaching of the antibody from the material?
Well, that it's possible since we are only using physical adsorption. Actually, we made a kind of test with an antibody, a different antibody that had a fluorophore. And we made a lot of washing steps, and we saw that in the beginning, we lose a little bit of the antibody, but after like two washing steps, the luminescence from that antibody remain fairly constant. So we believe that the antibody is still attached to the surface. However, we know that this is not the most accurate process. It was the physical adsorption. We use it because it's simple. But we want to try covalent bonding because in this with this physical adsorption, we are not sure if the antibodies are aligned in the way that the proper alignment. We cannot assure the proper alignment of the antibodies to react with the analyte. And our next step will be trying the covalent binding.
Great, thank you. And just one curiosity, and if I may, I think it's only Francisca with a raised hand. And I already spoke about this with you and Professor Florinda in the past and regarding your project and within your project. What is your opinion, and now that you saw the presentation of Matheus, because we already discussed this, now in integrating the aqueous biphasic system concept presented by Matheus and your zinc oxide transducer platforms for biosensing. Do you think that they can be integrated? I mean, do you see any possibility of having the material or having inside that aqueous phase where we have our target biomarker?
I think so. Not in the platform that we are using here, where we deposit a sample on a physical substrate. Maybe we can place our nanoparticles, our nanostructures into your solutions, and that way try to make the detection. I believe that can be done.
I do hope so. I wish you
Me too. Me too.
So thank you so much, Joana.
Thank you.
Okay, and now we have a question from Francisca Silva. Francisca, please. Microphone. Francisca, turn on your microphone, please.
Sorry.
Okay.
I thought it was already on. Okay, thank you both for your very nice presentations. I have two questions, one for each. So first, Joana, I would like to know if you have think about testing like multiplex quantification of disease markers, because one of the biggest challenges in diagnosis and even prognosis is that sometimes you need more than one marker to indicate the stage or the level of the disease. And is this possible with your approach? Have you tried some kind of these studies?
Well, thank you for your question. Actually, this is one of our goals, but we are far from that yet. We believe that it's possible if we use different bioreceptors in our samples, but we have to check if they lead to different signal changes in our transduction. Because if they lead to the same change, we cannot discriminate if we are detecting one thing or the other. But this is one of our goals, but we are not there yet.
Okay, thank you. And for Matheus, my question is regarding the systems that you use, and you use systems composed of ionic liquids and salts, and with very high amounts of salt. Can you substitute this type of phase formers with others to improve the separation, the selective separation of more than one biomarkers in your system?
Thank you for your question. And actually, it depends of your goal, because, for example, if you are just looking for a depletion of human serum samples, you can choose a system that will induce this kind of behavior. And if you are looking only for the extraction, for example, in the system that I applied for urine samples, you choose one kind of system that don't need to induce this kind of behavior. So it depends of the samples that you are studying and the goal that you want to achieve with your system. So, for example, we already know that in the literature is already reported for systems that induce an interface, a solid interface. So this kind of systems is better for when you have a high amount of proteins in your sample. And if you don't, you can use systems that don't have this kind of behavior. So it depends on your goal and the sample that you need to make a pretreatment.
Okay, because my question is because you have very high concentrations of your phase forming components, so you have a low amount of water, and sometimes the viscosity can be a limiting step for further application or advance in the field. So if you had some remarks on that aspect.
No, actually, this kind of behavior for the proteins to be in a solid interface is basically in a combination of effects in the lower amount of water that we have in our system. So we have a strong salting out from the salt, and also the higher interaction of ionic liquids. So we have some kind of a punch in one side and punch in another side. So the proteins are in the solid interface. So the only experience that I have with changing these kind of mixture points in the ABS was in the serum pretreatment, and we saw independent of this the ionic liquid amount or the salt amount in the biphasic area, we have this behavior. But in the urine samples, we didn't test it because the only goal was they achieve the higher concentration of PSI.
Okay, thank you.
You're welcome.
Okay, I don't see any further questions. Joana, you didn't say much about the, let's say, the material science side of things, which of course is something I'm interested in. When you make your rods or tetrapods or whatever it is, zinc oxide, do you have a narrow distribution of particle sizes? Do you have a wide particle size distribution? Does that matter in terms of the response?
Well, it depends on the conditions that you use to grow the samples. In the conditions that we use here, we have a wide size distribution of the branches of the tetrapods. But this is something that it's also beneficial in this case, because we want to have a high surface area. And with the smaller tetrapods filling the voids between the branches of the bigger ones, we can have an enhanced surface area. So that is a good thing. But actually, with this technique, we can control more or less the size of the samples that we are producing. However, in this case, since we are using high powers to have a higher rate of the growth, we have a higher gradient inside our chamber. And since we have this gradient, we will have very different sizes.
Yeah, but my main question, in fact, was having a wide particle size distribution, does that bother your signal? I mean, is that good or bad for your signal?
No, it's what I thought I said before. Since we have this wider size distribution, we have the small tetrapods that can fill the voids that are somehow made by the branches of the bigger ones. And
yeah,
in this way,
if you only had small particles, you would not need them.
Yes, that's true, but it doesn't make a big difference in the signal outcome that we obtain.
Okay. And Matheus, I'm always worried when I see something 100%, and all your slides have 100%. What's the meaning of 100%?
100% meaning that our system was able to extract our target protein for just one phase. And I heard this comment, at my PhD defense, and one of the jury panel, they asked me why my results are always 100%. I don't know if it's lucky. I don't know if I am a good worker, but in the systems that we apply, I have, I don't know, maybe lucky to find one system that was able to extract, but we also have a possibility to apply in a different kind of systems that possibly will have different results. So the systems with a combination of ionic liquid and salt was the perfect match for this application. I think that I found the perfect match between the two phases, and then possibly if I applied for a different ionic liquids or a different salt, the result will not be the same. So
one day you were sitting down trying to find out the right salt and the right ionic liquid, and then no. Actually, when I start this work in my PhD, I make I use different types of systems to understand the behavior of these systems in proteins. So I studied polymer-based ionic liquids, polymer-based ABS, ionic liquid-based aqueous biphasic systems, different types of systems to understand which one was better for my application. And then, for example, with the results that I had, that I didn't show here, but I use it as preliminary results in my PhD thesis, I found that, for example, polymer with the same salt, changing the ionic liquid for a polymer, I don't have these remarkable results. So using a polymer, I don't have 100% of extraction. So using ionic liquid, I have. So this is why I choose to apply for real samples, ionic liquid-based aqueous biphasic system. Okay, Matheus, thank you. We passed the time in 2 minutes. May I congratulate all the speakers in the first session, João, Joana, and Matheus.